Um, no one here today in ICHSSE questions the importance of history in science education. But for us, most of the problems and the challenges have been persuading others, developing curriculum materials, guiding teachers, producing assessment instruments, and ultimately integrating history among the institutional goals and daily practice. Well, that has been the story largely since 1947 with Conant's work on case histories in science. In the past several decades, things have changed significantly, particularly with the publication in 1996 of the National Science Education Standards in the U.S., which designated history and nature of science as an essential part of scientific literacy. That has expanded globally, and now nature of science is a major component of almost all international curriculums. Ironically, however, the history that was important to the philosophical views that are now being incorporated has largely been peripheralized and left abandoned. NOS today has taken a slightly different turn, and institutionally, the emphasis is now on inquiry, involving students in their own practices and reflecting on those practices about how science works. So our strategy, if we are interested in promoting history more broadly, I think is to try to tap in to this trend in inquiry and to integrate history and inquiry, which was the theme of ICHSSE in 2014. History and inquiry. Even more perhaps, history, we can show, can improve the inquiry for other teachers. For example, history can help to motivate the students to be involved in the investigations and the questions, both through a cultural context, as we've just seen, and through human contexts. History can also help to problematize the questions that lead to deeper reflection about how we produce knowledge or the nature of science. Historical cases in particular excel at being able to reproduce the uncertainty of science, or what Bruno Latour called science in the making. And this is basic to understanding that our conclusions are drawn from the evidence and not the other way around. The history also provides a convenient lineage of questions which helps to structure inquiry over time through an episodic or interrupted narrative. We should not neglect the fact that the narrative or storytelling format itself is a very powerful way of communicating for people who are not trained as scholars. This is how we communicate in our daily lives about what happened and explaining how things came to be. All these elements should be of interest to the inquiry teacher who is not yet interested in history. The use of history we can show them will benefit by highlighting these links between history and inquiry. Today I'd like to share a few examples of this strategy that were developed by some students that I worked with at USP 3 years ago. Framing historical episodes as student inquiry cases, or as science in the making. Each contains a series of inquiry questions motivated by and structured by a historical context. and threaded together by a historical narrative and designed to foster reflective nature of science thinking. These cases that I have to present today also exhibit another feature that is attractive to most teachers, which I think will further enhance the chances that history is adopted in actual classroom practice. And that is, they help to celebrate local scientific achievements. Here we are in Brazil, and the 5 cases I've discussed today all involve science in Brazil. And fortunately for the Brazilians, these cases are also written in Portuguese. Of course, this local orientation is not essential to understanding the nature of science, but for the teachers who we are trying to convince, this is often what they will use to decide what happens in their classroom. National pride and the prospect of providing local role models for students, which are powerful motivators. So, let me introduce these historical inquiry cases and some of the nature of science elements that they help convey. The first case is about Vital Brazil, who discovered the specificity of snake venom and went on to establish the Butantan Institute, now one of the world's leading research institutes in immunotherapy and animal toxins. The first feature of historical inquiry, you will recall, is the cultural and personal context for motivating investigation. In this case, we situate ourselves in the 1890s. Nearly 3,000 persons a year in Brazil die annually due to encounters with venomous snakes. Many are illiterate immigrants working on coffee plantations or building the railroads that help to transport the coffee. Few doctors are available in the rural areas. Workers relied primarily on local healers and their herbal remedies. But are these remedies truly effective? One motivation, then, for doing science is, can we heal snake bites and do so effectively? We also add the personal element, in addition to understanding the danger of snake bites, by looking at an individual physician, Vital Brazil, who is working in the town of Botucatu in São Paulo state. He is very interested in systematically testing those alleged treatments. As we move forward, we want to focus some of these problems on particular nature of science elements. So we focus the question, we choose our questions appropriately to bring those issues to the forefront. In this case, to test the remedies, Vital Brazil needs test animals. Well, fortunately for him, dogs and goats and guinea pigs are readily available. But what about securing the snake venom? Hypothetical snake venom, written in some kind of reasoning prose, doesn't work. You need living creatures, along with their real dangers. And Brazil, at this point, is deeply afraid of snakes. So, how will you extract the venom? This is Vital Brazil's challenge, and it is your challenge as students to work on this problem. This is a particularly nice problem because while we might go to Wikipedia or Google to find the answer, we are situating the students here to work on this themselves. They do not have these resources. There's uncertainty. This is science in the making. They must create the solutions themselves. After the students generate and discuss their responses to returning snake venom, we turn back to Vital Brazil. He works with the local healers to get the snakes. Living snakes, might add, that have been captured rather than immediately killed because of the threat. The first two specimens are mishandled and soon die. And suddenly you realize that if you need snakes, a seemingly trivial task like the proper handling of snakes can ultimately be essential to the practice of science. The third snake survives, however, and Brazil presents his snake with a fake target, a small mass of cotton, into which it releases its venom. Much later, the director of a research institute helps develop a long-handled device for lassoing the snakes, which is now known as Lutz's loop. Now, with all the supplies and specimens in hand, that was the hard work. The task of administering the remedies and comparing them is relatively simple. But note the nature of science lesson here. Science needs concrete and sometimes risky work in the material world. It's not just about theories or reasoning or imagining ideal experiments on paper. Of course, the investigation is not complete. But where do you go next? Student-led inquiry can be somewhat blind and haphazard if they are working on their own. If it is too preordained by the teacher, the students feel manipulated and simply wait for answers. They lose the core lesson of uncertainty and of science in the making. So, the fourth feature of historical inquiry is to follow Vitor Brazil along a lineage of questions that emerged naturally in a narrative format. When Brazil finds, not surprisingly perhaps, that none of the local remedies are really effective, he consults elsewhere. He reads about work in Europe that indicate a role for anti-sera, a new approach to medicine. In this case, you immunize animals to snake bites, then extract some of their blood, and use this to treat organisms that have been bitten. Now, the dominant theory ranks the toxicity of various snake species, meaning that the sera from the most venomous snakes should work against all. Brazil is eager to get some cobra venom, the strongest. But it all comes from France, and it's very expensive. Well, when he finally secures a sample and tests it, he finds that it has no effect at all on the Brazilian snake bites. So now, apparently, he will have to make serum from Brazilian snakes in Brazil. That needs a fancy lab. Brazil finds a new job and relocates to a bacteriological institute in the capital city of São Paulo, where the director allows him time and resources to continue his interest in snake bites. He begins collecting several species of poisonous snakes and immunizing animals to develop anti-sera. He looks for the strongest venom, but the results are unexpected. And here now it is the student's turn again to examine his findings and to interpret them. The rattlesnake antivenom works on rattlesnakes, but not on jacara venom. Likewise, the jacara serum works on jacara bites, but not on rattlesnake bites. Students are generally able to conclude, along with Brazil, that you need serum from the snake that bit you. The snakes are specific. They are not arranged from weakest to strongest. Now, at that time, that discovery of immunological specificity of snake bites had global significance, changing how immunologists conceived poisonous venoms, and not just in snakes. The students thus are able to share indirectly, vicariously, in this very important discovery. And that's historical inquiry feature number 7, the role of an experiential narrative or storytelling format in helping to convey an abstract idea. So, to prevent snake bites, Brazil needs to make sera for each snake type. More snakes, more lab resources. But for a while, Brazil is prevented from doing much. Plague has broken out in the nearby port, and his job is to make anti-sera for plague. That takes priority. But it all proves a benefit eventually. To be able to produce large quantities of the plague anti-sera, the government institute purchased a large farm outside the city to maintain the horses and sheep that would function as organismal factories for the serum. When Brazil resumes his work on snake venom 2 years later, he now has all the extra resources he needs. But now Brazil needs even more snakes and a place to keep them all occasionally while harvesting their venom. Another problem for the students, part of this historical lineage of questions. Where do you keep snakes safely? Brazil constructs a large serpentarium surrounded by a pit and sheer walls. In time, this small snake zoo becomes with its thrilling proximity to danger, becomes a popular attraction for those visiting the site. At the same time, Brazil creates an exchange system for keeping an adequate stock of snakes arriving, and then to distribute the sera. When workers in rural areas ship him a snake in one of the heavy wooden boxes that he provides, the Institute provides in return several bottles of serum and syringes for the treatment of the snake bites. Establishing that system helped transform an ideal of potential treatment, theoretical treatment, into a sustainable reality. Mortality rates from snake bites dropped from 25% to 2%. This is science in action, and it exhibits substantive work and resources, not just abstract scientific reasoning alone. Vital Brazil works over the next several years to transform the farm into the Butantan Institute, which has become today one of the most important worldwide research institutes in production of vaccines, anti-sera, and other biopharmaceuticals. So here we have historical inquiry feature number 5, happy ending, and a resolution to the problem. The original scientific problem of healing snake bites is solved, and the personal and cultural story comes to a satisfying end. This then becomes an opportunity to review and consolidate the learning about the nature of science. In this case, we have seen repeatedly that scientific ideas are one thing, but without the institutions and the resources that materially enable research, science is empty. The image of the serpentarium, in particular, which has become a popular tourist attraction, is celebrated in postcards, helps to underscore this indispensable role of infrastructure in science. So, that is the first case of Vital Brazil, a scientist of international caliber, but also a Brazilian hero. It is also a story of science solving Brazilian problems. The local connection can make the science seem more relevant and more important to the students, and so contribute to the core problem the teachers face of motivating and engaging their students. Let me turn now to a second case, which also illustrates the historical inquiry model and also involves a case of disease and health. Now, this one involves a scientist that most Brazilians know by name, and so the case is written to keep his name hidden and obscured until late in the story. The questions and the unknowns are foremost to revive this sense of science in the making. So we cannot give a clue or spoiler that will prevent them from experiencing that form of science. The initial problem, as I noted, is a disease among railroad workers. Early last century, a young physician is sent to the countryside to treat the patients. At first, the disease looks like malaria, but also not exactly like malaria. So students are given a list of the symptoms of each disease, and they must decide for themselves. Is the disease merely a variation of malaria to be treated the same way, or is it something entirely new, which needs a different form of treatment? Further observation indicates that in later stages of the disease, the patients develop these enlarged thyroid glands. So now is the disease a form of goiter? Again, the students receive the symptoms of each and must decide for themselves. At the same time, they must begin to develop what criteria one uses to help differentiate one kind of disease from another kind of a disease, a much larger question. Well, while visiting a more remote district, our physician hears from the chief engineer of the railroad that many patients with the goiter have problems with bed bugs in their shacks. He wonders if there might be a connection. Now students face yet another problem: expertise. The engineer is not a doctor. Should our physician trust what he says? Well, it turns out that just a few decades earlier, Carlos Finlay had identified mosquitoes as carriers of yellow fever, and our physician decides to investigate further. But now again, you must decide how you are going to research this. Here the students must think out their own research strategies and organize a search, realizing that you must often rely on models, analogies, and hypotheses to be able to structure your research rather than just blindly looking and hoping that a cause announces itself. Along with his colleague, our physician collects several samples of the bug. Following the example of yellow fever, as his model now, he looks in the gut for microorganisms and finds there some flagellated protozoa, which he identifies of the genus Trypsinoma. But is this the cause? Because of his interest in diseases, our physician examines the blood of other animal species and finds another form of Trypsinoma in the local marmosets. But he cannot be sure if it is exactly the same species as the one that he found in the diseased bugs. Oh, we lost our marmosets. And the students again then are challenged to propose ways to investigate this problem. Are the two forms of Trypsinoma the same? Are they both causing the disease? In this case, with the help of a major research institute in Rio de Janeiro, the team finds that when marmosets are infected by the bugs, they develop the new disease in 30 days, and they show signs of the microorganism in their blood. But now they find it to be a new species of Trypsinoma, unlike the ones that had been found in the marmosets earlier. So now our physician examines blood of his diseased patients, but they do not show the presence of the bug's Trypsinoma either. Apparently, with all this work, there is no connection. So, what do you do next? This is another dilemma for the students to solve. Well, in this case, our physician, Carlos, returns from the Research Institute to treat his patients. One young patient is severely ill in a very advanced stage of the disease, and he decides that he will take another blood sample. And the sample now shows the new species of Trypsinoma. So there does seem to be a connection after all, but the microorganism must have changed its form in the patient as the disease progressed. In the remainder of the case, the students learn about the changes in the morphology of the Trypsinoma and they see how the microbe moves from organism to organism via the bug, eventually to humans. The discovery of the new disease is confirmed and named after the physician that many people here will know, Carlos Chagas, a famous Brazilian figure, again, that they all know, but usually only quite remotely. In this case, Chagas is the first person historically to identify a new disease and to track down all the stages of the infection cycle, involving the microorganism, the insect vector, and several of the natural hosts. So there are several aspects of nature of science here involving economic factors, personal motivations, analogies between old diseases and new diseases, local knowledge versus systematic research, the role of chance or accident, and interactions of laboratory studies and field studies. I hope this case further illustrates the role of a lineage of questions, the sequential episodes of inquiry for students. They follow the history of a major discovery, but in small steps. The students thus participate in authentic science, not artificial or cookbook classroom investigations. And the answers for them at first are not obvious. They must work with the uncertainty and make the conclusions for themselves based on the evidence available, rather than knowing or guessing the right answer. Now we will remove from our script because the paper printed out is incomplete. So this is Chagas' great discovery. So now we want to shift to a third case to benefit the physicists. We're tired of hearing about biology, perhaps. This is a case involving a well-known episode in the history of science about the testing of Einstein's theory of relativity. Arthur Eddington, a Quaker, is interested in promoting peace and doing so by promoting a German scientist in an English population. So he decides he wants to test a prediction of general relativity, the bending of light around an astronomical source from gravity. On Earth, this can only be observed during a total solar eclipse, when you might see the difference between the observed position of the star and the one that you know that it exists in on normal occasions. Well, this is happening in 1917, and there is a solar eclipse coming up in 1919, and the first task for the students is to decide, well, where are you going to go to make this observation? Here they have the path of the eclipse, which that can be predicted, passing through Africa and partly through Brazil. Eddington, in this case, consults an astronomer in Brazil, Henry Moritz, and asks him what an appropriate site might be. The students, of course, must begin to develop the criteria about what a site must accomplish. For example, it must be in or very near the path of the total eclipse, so you can maximize the time of observation. Cost and budget is a factor, so it needs to be near a major city and port to transport all the equipment, both the telescope and the photographic equipment, and all the astronomers who will be in attendance. Eventually, Moritz recommends a site, as many of you know, in Sobral. And on a date in May in 1919, they all assemble at the site to watch the eclipse, hoping, of course, that the daylight and the clouds will allow something during the middle of the day. They have shielded the telescope from the sun, but perhaps not well enough. When the photographic plates come back, they find that the stars are not where any of the theoretical predictions place them, either where Einstein says they should be, or where they should be if there is no bending at all. And the students must consider what to do if the predictions are not correct. Do we throw out the data? Do we look back at the experiment? People begin to suspect that the heat had distorted the lens and changed the image. Can you fix that, or is that cheating? And that's still a problem which is considered today by many philosophers. So, this is a nice case of considering institutional aspects and also error in science and the role of a Brazilian scientist in another very important 20th century discovery. Let me move now to a fourth case, someone who is probably less well known to people here. Joanna Dobereiner. Her work begins in the late 1950s and extends over several decades. Having established a role for nitrogen-fixing bacteria in many tropical crops, she's looking at nitrogen-fixing bacteria in many tropical crops, in addition to those that have been known in legumes for a long time. Dobereiner arrives in Brazil in the 1950s with her husband, an emigre from Czechoslovakia. At first, she, and the students following the case, must decide if, despite her credentials and experience, she will accept an unpaid position for working. Ultimately, she does accept, allowing her to continue her previous work on soil microbiology and agronomy. When her supervisor leaves, she is now left to work on her own. She reflects, in particular, on the problems of fertilizers, which are now promoted by most agricultural models from the U.S. Joanna notices that some strains of plant develop even if nobody fertilizes them. Bahia grass grows everywhere in Brazil without any fertilization, and sugar cane productivity seems to remain constant for several years without the use of nitrogen fertilizer. Again, taking into the position of Joanna, students speculate on the possible reasons why these plants can survive under these conditions. With her background in soil microbiology, Joanna considers the role of independent nitrogen-fixing bacteria. One such species is Azotobacter. She can test their growth in Brazilian soils. But what factors here might be relevant to the investigation? Students must decide. What are they going to investigate precisely? Dobereiner considers temperature and conducts climate-controlled experiments. She concludes that higher temperature matters to Azotobacter growth. The numerous studies already done in Europe and America in temperate environments turn out not to be relevant in Brazil. Joanna also considers the role of acidity. Azotobacter, researchers have claimed, do not like acid soils, but these are what are common in the tropics. Joanna finds a previous study where they are found in some tropical soils. The students receive the researchers' data on the percentage of Azotobacter in soil samples with different pH values, and they are left to interpret any patterns and compare their conclusions with those that are published. Joanna, in this case, must reject the widely accepted conclusions that have been published. She decides that they are not informed by studies in the tropics and that they are biased by the theories that the scientists prefer. So, based on lab studies, Azotobacter should be able to grow in Brazilian soils. But do they? Students must then plan now again how to proceed. Joanna must examine soil samples from very different geographical regions in Brazil. She conducts more field experiments based on different soils inoculated with Azotobacter. They must be tested again with different conditions of temperature and acidity. And she finds that yes, they do add nitrogen to the soil, enhancing the growth of many plants, then potentially crops. Meanwhile, some of the Azotobacter species are reclassified in another genus, Bergerinca. This opens new possibilities for other related species that have not yet been considered. So, more repetition of the same kind of work. Joanna takes a break in her work to finish her formal degree, traveling to the U.S. And she returns to Brazil and with her new degree is appointed to the National Soybean Commission. Economic conditions favor the export of soybeans. But here is the next question: what can you do to increase production? The dominant model of agriculture is to develop improved genetic strains and then make large capital investments for mechanized farming, using substantial inputs of fertilizer and irrigated water. Joanna considers instead the role of biological nitrogen fixation. Soybeans are not known to have a symbiosis with rhizobium, but Joanna is skeptical, and she searches for several other strains of rhizobium and finds ones that do interact with soybeans. At first, her work is largely disregarded because it is irrelevant according to the normal agricultural practice. But as the difficulties and expenses of the capital-intensive agriculture increase, Joanna now has an alternative to the costly use of fertilizer, which makes soybean production cheaper even than in the U.S. Soybean production increases and continues to do so over the next several decades. By 2003, Brazil becomes the second largest producer of soybeans in the world, accounting for over 1/4 of the world's production. Joanna's attention to the microscopic bacteria in the soil have resulted in major economic benefit for Brazil. And of course, it is another example of science in Brazil. There is one other case that I would like to mention very briefly, and that involves Theodorus Dobzhansky. Dobzhansky is a renowned environmental biologist who worked to help establish the modern studies and its connection to population genetics. He is invited to Brazil on an occasion, comes, and decides to study the wild fruit fly populations here in Brazil. In several visits over many years, working with colleagues here, he collects important information on the genetics of involving populations. At the same time, he helps to establish the genetics department at the University of São Paulo, another important institutional framework for doing science. Well, these cases that I have described all feature science in Brazil. That should make them, I hope, attractive to Brazilian teachers who might be encouraged to teach more about history and about the nature of science. Of course, with a local or regional focus, it is easy to overstate the importance of some scientific discoveries, and thereby, in a sense, dilute the authentic lessons about the nature of science. But these cases were selected precisely because the discoveries were of global significance. They are just as valuable in the US, Canada, Germany, or elsewhere, where they can help profile the global nature of contributions to science. What matters most is that the inquiry problems are significant, the narratives compelling, and the nature of science lessons clear and insightful. Of course, such studies are also vulnerable in another way. A common tendency in telling such stories is to amplify the heroic and nationalistic elements, creating monumental and ultimately unrealistic images of scientists. That is, they may easily succumb to myth conceptions, pseudo-history, and unfruitful rational reconstructions, a theme that I presented at the fourth ICHSSE way back in 2001. When this occurs, the historical cases actually fail to render the history of science, the nature of science, faithfully. So a focus on local and regional heroes needs to be balanced with healthy historical realism and with honest acknowledgement of the non-idealized elements of the nature of science, such as the dead ends and failures that we encountered with some of our stories. Nevertheless, when properly contextualized and enriched with authentic inquiry, I think these local historical cases can offer an attractive opportunity for teachers that will engage students in lessons about the nature of science. I hope this proves valuable, and I am very interested in opening discussion to what other people think about the nature of local and regional discoveries and their experiences in doing so. Obrigado.
Questions? Okay.
Thank you, Douglas, for the brilliant speech. I have two questions. The first one is more pragmatic. Which resources you think should be available for the students to tackle these questions that you interpose during the narratives? And the second one, you addressed that at the end of your speech. I mean, by focusing the stories...
Let's do these one at a time.
Okay.
The inquiry questions are central both to regular inquiry and to historical inquiry. And this involves the teacher knowing the caliber and level of their students. These cases can apply at various levels with the level of difficulty of the problems based on what the students already know, floating to more difficult with advanced students and easier, giving more clues or more background information to help the students to nurture them along, but still engaging in uncertainty and questions. Sometimes, I think you were asking, sometimes should they use historical data? So this is going to be a choice for the person who writes the case study to know if it's available, if it's easily understood, if it's a good table or chart form, or whether it needs to be a graph, needs to be regraphed. So this is in a sense a discretionary decision by the individual teachers and by the writers who know about the historical material and what is available and what might be fruitful for students, imagining they are writing for a certain level of student. Yeah? Okay.
The second question, you have addressed that very rapidly at the end of your speech. I mean, by focusing on individuals in the story to tell these narratives, how high is the risk of losing the collective dimension of sciences?
So there is perhaps something of a trade-off. Having a central character makes the storytelling and the narrative stronger because you are following one character and you identify with them. This does not prevent you, of course, from bringing in other individuals, co-workers, collaborators, people who disagree and create debates. So the fuller versions of these cases indicate more of those. They talk about the Chagas work with Belisario Pena, for example, and you get more about the nature of the railroad engineer and the people who were at the research institutes that they all depended on. And these in turn can become occasions. There's one case in the Doberreiner case where she must decide whether or not she is going to collaborate with another scientist. And is this a good idea or is this just going to make things difficult? So you can focus these things, and then always at the end of the case, when you review the nature of science elements, you pose this question: how important was this one individual compared to the others? And the students, by hearing the narrative, will go, important, but not only important. Other people are important too. So you can create the case study to bring in the other people, and if you want to underscore. So in the Eddington case, for example, the whole expedition was conceived as an effort in international cooperation. So you get more of this social element when you focus more on Eddington and the before and after, and not just the parts that happen in Brazil.
Thank you for your talk. I think it's very tricky to deal with national pride, and there are some dangers. And in those cases, first, Vital Brasil. Of course, that has never been my research subject, but once I was invited to talk about him by one of his grandsons at a conference.
Very awkward.
And I'm sure they will never invite me again, because I did my homework, and for some weeks I researched things, and things are not as beautiful as they seem. In the case of his research, it's necessary to take into account other researchers that were doing a study on snakes, not only in France, but also in Brazil. There is a guy who worked at the National Museum in Rio de Janeiro, João Batista de Lacerda, who was trying to find some chemical way of dealing with the serpent bites. And that's very strange, but no one tells that when you apply the serum that was developed by Vital Brasil and other people, you save the life of people. But there are side effects that are terrible. And if you see the hands and arms and legs and feet of people who have been bitten by serpents and treated, it's terrible. And it's very curious that the chemical treatment that was developed by João Batista de Lacerda does avoid those side effects, but the two methods were never combined, and it's a problem that remains after more than 1 century. So I think that we can't focus only on positive aspects and on those national heroes. It's very dangerous. In the case of Carlos Chagas, I think any historical study must take into account the criticism he received at the time, especially from Argentinian researchers, and he couldn't reply to the criticism at that time. Of course, from our point of view, we say, but Carlos Chagas was right, but he couldn't answer the criticism at that time. So I think it's quite dangerous to choose those scientific heroes to deal with.
Okay, so I agree completely that we need to avoid presenting only the positive aspects. leads to the myth conceptions and to not understanding science correctly. To the degree that these are ways to get to teachers, to get them to think about history, they might begin with these histories and then lead to others that are not just necessarily national heroes. So I think that it is important to consider the benefits that they provide, at the same time cautioning against this over positive influence, and also perhaps changing our notion about what a hero is, and that a hero must always be perfect. I think we can celebrate the discoveries of all these people and still acknowledge that they were flawed. Perhaps they were very cruel to their lab assistants, or they made ethical shortcuts to be able to get to their research, or they did not deal respectfully with their colleagues in this nation or around the world. I think this makes them more human, but perhaps not less of a hero. So I think we should think about and rather than not. The hero and the flaws rather than no heroes at all. And this problem that you pose underscores, I think, the importance that when we construct these case studies, particularly of the heroes, we use good professional historians of science who know the details, who insert the qualifications and the errors and the flaws and the unpleasant aspects that we don't want to always acknowledge, but to ensure that they are included in the case study, because the people who are not the historians will only tell the positive story. So this is partly then a recommendation about we need to be very careful about these case studies. Take a lot of time, but they are worth investing in, I think, but we need to be careful about who we get involved to make them, to write them. Did Newton do work in Brazil?
Claro did come to Brazil en route to Peru, but that's another story. Thanks, Douglas. Speaking from the perch of North America as a man,
Inherently inferior.
Inherently inferior. It's a struggle for me to see the relevance of talking about, and we talked a bit about this the other night, Brazilian science versus just science, right? On the other hand, though, when Augustine was up there, I was waiting for him to talk about Rutherford having been in Canada. And that didn't happen. So it's an internal struggle of mine that I'm sort of airing out my laundry here. But in response to, to the last comment, it would seem to me that your examples offer yet another opportunity for the teachers to navigate through all this. And as much as I like inquiry, I wouldn't teach this using inquiry. I would teach this using problem-based learning, because it seems to me that you have there is a difference. It seems to me that you have these wonderful vignettes, if you wish, and you come up with these wonderful questions and then throw those questions, and now the students have this problem. Okay, so what do I need to solve? Sorry, I forget all the examples except the Eddington one. So what do I need to solve the issue of a solar eclipse? So now they go off, they do that. They come back and they say, oh, wait a minute. We've got to have this location, we have to have all this material, and now we need to get local people involved. Because so now what does it mean to get local people involved? What does it mean to do this in a country that might not have an infrastructure, right? So I'm thinking a PBL approach might actually be a way of approaching the history of science in a localized or regional context, and wondering if that makes sense to you.
We should have a more extended discussion about what problem-based learning means, what case-based learning means, what inquiry learning means, what historical inquiry means. And part of this might be the scale or scope of the question that you throw at the students. In other words, you might give them the whole problem and just expect them to do the whole thing. Or you might take them stepwise through it, giving them tasks that are easier to organize at the level that they are in school. So, and again, how much background you give to the students is going to be a matter for the person who's writing the case study to consider. What do they need to know to be able to answer this, but in an open-ended fashion that doesn't prescribe only one answer, the right answer, which it can be a problem in historical case studies, which we've seen from studies in Germany. It's a balancing act. Yeah, is that fair?
Is it fair? Yes. In fact, I think we agree now because,
Okay.
to me, if you want to raise some important questions, okay, what are some of the serious effects of snake bites, or how are women in science treated? So she's asked to lead a lab without any resources. I mean, we have other historical examples that's important, and so depending on the level of student, the grade level, and so on, you could structure the case with more detail or less detail and have them explore. And that would be a way of managing the not so clean parts of science in the making, I would think.
And in a larger view, I wanted to contextualize many of these cases in the large-scale institutional problem of getting history inside the door of the classroom. Once we do that, then we can start bringing in our other case studies. We can start making more complex case studies. We can start scaling them. Simple case studies when they are younger, more complex when they are middle-aged, and as they advance through school, giving them much more complex nature of science problems and complex issues that are that philosophers themselves will discuss. So again, I'm inviting reflection on, is this a good way in the door?
I think it's very interesting to have this look to the famous cases of history of biology, agriculture, etc. And but I've learned about something called pseudo-history. And I wonder if this kind of case, they are what they are different from the very traditional story of Newton did something, or the big Galileo, or the super Einstein, because this case that you presented somehow, at least Vital Brasil, Carlos Chagas, Eddington, they are already on textbooks, on science textbooks, and they have a simplistic approach that portrays the Vital Brasil, Carlos Chagas as heroes. And all the complexity of nature of science they are lost. The complexity is lost. And I think one of the big achievements of our community is to be aware that this is a problem. And one of the biggest flaws is also to be to develop, or no, it's not a flaw, but it's challenge to put the complexity of nature of science into the cases. And I think when we look to the textbooks and we look how these cases are portrayed, the complexity of nature of science is lost. So somehow I agree with the necessity of being cautious about that. And I think it's not so complicated to include the complexity in your cases, because I like how you write, how you tell the story very much. So it seduces the students. So the style is very well structured, and the idea that you can take lessons from nature of science, from the narrative. But I think it's necessary to include not only the flaws of the scientists, but also the complexity of the discussion, because there were alternative alternative views, as Roberto mentioned about Vital Brasil. There are more than just a linear story, you know. And since you have this talent for writing, I think you should think about adding more to the cases, adding the complexity to the case. And there are journals specialized in history of Brazilian medicine, Brazilian science, and I think it could be helpful to include the complexity in these cases.
Okay.
It's a comment. It's not a question.
No, but it's a valuable comment, because in part the problem is not just about the famous individuals, it's about all the case studies. They all have to have sufficient complexity to be authentic and for the students to learn something that they don't already know or think about the nature of science. So here is my perspective to consider. What are you going to do to get rid of the bad stories that are already there? You don't want them there, yes? They are a problem. And in a way, perhaps the best way is to provide a substitute that's better, that has more of the complexities, that involves some of the struggles, some of the more human elements that make the people more human, less godlike, that indicate that it was not simple, that it was difficult, that there were people who disagreed and how that unfolded, that this is part of what these good case studies, more complex case studies will do, engaging the students through that whole process. And I'll note that we got, of course, very abbreviated descriptions of these cases. We did not get the whole case. So I'm presenting just the tip of the iceberg to make us aware that these cases are available in Portuguese, written by Brazilians. On top of that, I helped, but they did the writing. So I think substitutes that are more sophisticated than what is currently available is a better strategy, possibly, to reflect on than trying to complain about the fact that, oh, there are all these bad histories in there. What are we going to do about them? Provide an alternative.
I have a simple question. Do you have some examples that was developed in science class, especially the case of the Maurice? Because I'm curious, because Sobral is a very small city in the north of Brazil. I lived there when I was young. Yes, and I was curious if the students think about the local people involved in process. Yes, because have a problem with the adjustment of the instrument, and for some expedition that come from a city in 1999, 1990. Yes, 1990. And this local people is very small city, yes, and how they receive the expansion. So I become curious. The students think about that, because in the process, maybe they could bring a question that are not planned in their narrative, yes. It's interesting because when I lived there in 1970, this episode is something that are common to discuss now so in the city. Oh, in the past there are many strange men that come here and said that then the day become night. Yes, and the day become night. Yes, so for them, or for the local people, said, oh, it's very interesting, very strange. So I'm curious about that. If you have some example in science class that develop, no?
So I want to say all case studies are a work of judgment and art. So the team that created this case study, I wanted to make for them to emphasize something about the Brazilian connection, to make it a little bit more relevant. But they were hardcore physicists and wanted to focus on Einstein teaching relativity. At the same time, they were teaching the testing of the theory. And so the actual where is it going to take place was, for them, a very minor part of the whole case study. One could equally well write a case study that ignored Eddington and put him in the background and focused just on Maurice and how he dealt with people or with some of the other people, and expanding all that story to tell the story, which of course may be of much more interest to the people in that local region than it would be to the people in Sao Paulo or people in Paris or Singapore. But I think that's an equally legitimate way to write a case study. The work just needs to be done for the individual occasion. Yeah.
Thanks for your several examples. In your presentation of most or each of the examples, you, at least as the commentary on these cases, pointed out to us a nature of science kind of lesson that you felt that this case was presenting. And maybe that supports the rationale for us valuing that, at least in this room, and valuing that case. And then I think back to Andrea's presentation in her first example, the anatomy history of anatomy lesson, where the teacher of the class, in engaging the students with the painting, the Rembrandt, and the other one, the Vermeer or whatever, was the students raised questions and realizations that were powerful for them, that could be considered very significant culturally and nature of science like realizations, and which were not what the teacher expected. And so and this has happened for me. Whatever I think is going to be the major lesson, then I find something else happens. So I'm just interested in your kind of response to that, because as I guess these cases that you presented today haven't been presented in classrooms, or if they have, you don't know that part of the story.
The Chagas case and the Brazil case have been field tested, and there will be articles forthcoming in, I think, the end of this year that describe how they work in the classrooms. there are certainly ways to approach the nature of science that don't involve history. They might involve contemporary cases. Rather than going through ancient photos and images, we could start talking about what's happening now. The other major thing is engaging the students in inquiry of their own. Well, what are you interested in? Let's see, are there scientific approaches that will involve us, and you will do the work as your own small-scale scientist. So I certainly don't want to present that there is only one way to teach nature of science, or only one way to produce a historical case study. But to the degree, again, this is part of understanding that in the large-scale curriculum, nature of science, or understanding how science works and how we evaluate evidence, is a major part of scientific literacy. And I think for all of us in this room, we think it would be virtually impossible to understand that fully without using history. So trying to package this in a way that becomes used and does the job of teaching the nature of science, using the inquiry, using the history, and in this case, combining them to some degree, is one strategy. There might be other strategies, and I would argue that in the case of the women in the prison, we're not really teaching them about nature of science, because the ultimate lesson is not, for them, I think, about understanding and using that knowledge and interpreting anything in the future about science. It is about empowerment and seeing their role, and particularly their possible role in a scientific role. And that, of course, guides a very different kind of teaching strategy, for which the case studies that I provide here would be useless. I mean, they might be, in the sense that Joanna Dobereiner has a few gender struggles along her career. And you could emphasize these and point out, and particularly in retrospect at the end of the case, well, what do you think about this? You know, how would we solve this problem? And I know that there are other cases where the struggles of the individual female scientists, for example, working among males, to present that to the students. And what's interesting, I think, is in terms of, you cannot expect what the students are going to say, but by having framed a question a certain way, like, where are you going to get your funding? You've begun to underscore the problem of funding, and they have to walk through it, and whatever they answer, they're in a sense underscoring the problem of funding, because that was the problem that you contextualized for them and made for them. And then seeing how it gets solved differently, that's another layer of the lessons of nature of science. So, not an exclusive strategy, but one that gets you certain things, which I think we all want.
Okay, we need to close it this first time. We thank the Professor Allchin.